Beef Research School on RealAgriculture.com is brought to you by the Beef Cattle Research Council.
So basically the bacteria have genes within their genome that code for proteins that help inactivate antibiotics in a number of different mechanisms. And those types of mechanisms can include, they may degrade the antibiotic, so they may produce an enzyme that breaks the antibiotic down. They may develop a pump that pumps the antibiotic back out of the cell that prevents it from entering. They may develop a metabolic pathway which basically works around the antibiotics so that the antibiotic is no longer effective. And they can also modify the antibiotic by attaching a molecule onto it and thereby rendering it inactive in that manner as well. So they're pretty clever organisms. They have a number of mechanisms and different procedures that they'll use. Sometimes they'll use more than one procedure on the same antibiotic. There has been some studies going on, like we've conducted some research examining that ourselves. There's a bit of work going on in the United States as well. In Canada, we've probably done as much work as anybody in the beef. There's been quite a bit of work done in poultry and in swine, and that typically because some of the antibiotics that they're using in poultry and swine are of more significance to human health than many of the antibiotics that we use in beef cattle. But the majority of CPARS focuses on the retail sector, looking at isolating bacteria from meats and looking for whether those bacteria are antimicrobial resistant. So our work that we have done has been much more on farm, looking at administering antibiotics at known concentrations and then measuring the impact that that has on the development of antimicrobial resistance and looking at methods, so whether if we reduce the amount of antibiotics we use or we take the antibiotics out of the diets at certain times, how does that impact antibiotic resistance? Those are some of the questions that we've been addressing. Well, there's very few antibiotics that there's a huge amount of antibiotics out there that are available to treat infection. Very few of those antibiotics are actually included in the feed for cattle. Some of the antibiotics that are tetracyclines. Tetracyclines are widely used actually in livestock production. They're also used to some extent in human medicine as well. Tetracycline is one example of an antibiotic that can be included in the feed for cattle. It's approved for that in Canada. Other examples are sulfamethicine and tylosin, which is a macrolide. So in the studies that we did, one of the things we examined is we put in tetracycline at a couple of different concentrations. And most people, if you read in the literature or in the popular press, you'll hear, well, subtherapeutic antibiotics always cause antibiotic resistance. And that's, in fact, not always true. So for an example, in the study that we did, we put in tetracycline in the diet at a concentration of 11 parts per million. And in that study, we found that it didn't contribute to an increase in resistance. Now, if we increased it to 44 parts per million, then we saw an increase in the amount of tetracycline resistance. So there's what we call a threshold to resistance development, in that the concentration has to be high enough so that it poses some degree of a threat to the bacteria, and then the bacteria will go ahead and take the necessary actions to develop resistance to that antibiotic. So at 11 parts per million, the concentration wasn't high enough, even though that's still the subtherapeutic level, to induce that resistance response in the bacteria. Well, if we summarize, in a nutshell, the work that we did in the past, and in the past what we did is we looked at the use of those subtherapeutic antibiotics that are used in beef cattle production, and then we looked at the implications of those in terms of, do they cause resistance to any bacteria, induce resistance in bacteria to antibiotics that are really important for human health. And in a nutshell, the answer to that is no. We didn't find really that kind of relationship. Now, there's no doubt that if you feed an antibiotic, if some people ask the question, well, if I administer an antibiotic, does it cause antibiotic resistance? The answer to that is invariably yes, because bacteria respond to antibiotics by becoming resistant. And so yeah, when we fed tetracycline at that higher concentration, we caused resistance. When we feed sulfamethazine, we caused resistance. It doesn't matter what antibiotic you're talking about. There will be resistance in some population. The critical question is does that resistance also cause that corresponding cross-resistance to antibiotics that are really important in human medicine? And in the studies that we did, we found that that's not the case. So for example, we looked at about 10,000 isolates of E. coli, and we used E. coli. E. coli typically doesn't cause disease in most instances, but it's a very good bacteria to use as an indicator. So we use that as a method of sort of tracking what's happening with resistance. And one of the antibiotics that's very important in human medicine is a class called fluoroquinolones. And ciprofloxacin is a common antibiotic that is used in human medicine, is from that family. And when we screened 10,000 E. coli isolates from beef cattle, we didn't find a single isolate that was resistant to ciprofloxacin. So it just indicates that that we're not seeing that cross resistance in those cases to antibiotics that are really important in human medicine. We plan on stepping it up in a couple of manners. One is that we're moving to a category 2 antibiotic, which is the macrolides. So that's an antibiotic that could potentially have greater implications for use in human health, because there's some other antibiotics such as erythromycin that are routinely used to treat infections in humans. And that erythromycin is a member of the same family as tylosin, which is one of the antibiotics that we also include in feed. So, we didn't find much at those lower levels. We want to look to see, okay, we've got something that's a little higher level, maybe a little more important in terms of its implications for human health. What's the implications of using that in the diet? So that's the next question that we want to try to answer. The other thing that we're really interested in is that if you talk about antibiotic resistance, if we're looking at antibiotic resistance in terms of human health, we're really interested in those particular bacteria that cause infection. It could be a potential cause of death in humans. So those particular bacteria, do they acquire resistance, and where do they get that resistance from? And you can almost look like the genes can move around independently of the bacteria. So you can have genes coding for resistance in one species of bacteria. It could potentially be transferred over to one of those particular bacteria that does have serious implications for human health. And that's what we want to look at. So these mobile genes. So we can use techniques now. There's been so many great advances in the area of genomics and gene sequencing that we can actually go into an environment and isolate the DNA out of that environment. So for example, we plan to isolate DNA out of catch basins. So we'll take a sample of catch basin water. There's a whole range of microbes within that water. We can then extract the DNA from that sample, and we can sequence all the DNA that's associated with that sample and look for all the genes that code for resistance to antibiotics. So in this next study, what we hope to do is to look at how prevalent is the resistance at the feedlot within those types of samples. So we may even start with a sample from the pen floor as the first site. So that's where the concentration of the antibiotics associated with beef production would be presumed to be the highest. That concentration would then be presumed to be somewhat lower in the catch basin, and then in the surrounding fields, and then in adjacent watersheds. So we're going to track that through those systems and say, okay, once we get to the watershed systems and we're isolating the DNA from that, what does that resistance profile look like relative to the resistance profile that we saw right at the feedlot, where the concentrations of antibiotics associated with beef cattle production are the highest? You've got to test these things. If we want to look at the things that have that implication for human health, which is ultimately what we're most concerned about, we have to look for these linkages and see if they're there. And in many cases they may not be, but if you don't do the research and you don't do those studies that try to answer those questions, how can you conclude that, right? And that always leaves this then sort of grayness area about they see a lot of antibiotic use in livestock, and then it's automatically extrapolated to that has an immediate impact on human health. And in most instances, that evidence that makes that connection is not there.